May I begin?
Yes, you can.
Okay. So greetings, everyone. The topic of my presentation today is information extraction from electronic health records and EEG signals in the search of a potential biomarker for ADHD diagnosis. And I am the author, and my co-author is Shikhar Khurana. So first, I would like you all to understand EEG. I'm sure you all know that universally a 10-20 system is followed for the placement of electrodes, and it includes 21 electrodes. So what the 10-20 system is that first, two lines are drawn. One is drawn from the nasion, which is the bony prominence over the nose, and the other up to the inion, which is the bony prominence over the occiput. And a horizontal line is drawn across both the ears. And then, according to the distance which is marked, say 20% of the distance of the total distance from nasion to inion is the distance between, say, the central electrode and the FZ and the PZ. Similarly, 20% distance is again between CZ, C3, and C4. So this is what the 10-20 system approximately is. An electrode is represented by a letter and a number. The letter indicates the lobe it is placed over, say the frontal, temporal, parietal, or occipital, and the odd numbers represent the left hemisphere, while the even numbers represent the right hemisphere. So C3 is the central electrode representing the left hemisphere. The Z indicates the center. So what does an EEG record? EEG records the electrical activity over the surface of the brain. The electrodes for the same are placed over the scalp. An EEG voltage signal represents a difference between the voltages at two electrodes, also known as an EEG channel. EEG channel can be represented as montages. There are namely two types of montages, the bipolar montage and the referential montage. The bipolar montage measures the voltage difference between two consecutive electrodes. For example, the voltage difference between, say, F8 and FP2 or T4 and F8. A referential montage, on the other hand, measures the voltage difference between a scalp electrode and a reference electrode. A reference electrode is basically an inactive electrode. That is the A1 and the A2, which are placed over the ears. So a referential montage would, say, be one which measures the voltage difference between C3 and A1. So these are other different montages. We have seen different types of waveforms on EEG. On a normal EEG, we see four different types of waves: alpha, beta, theta, and delta. The alpha and beta are the higher frequency waves, which have a frequency of more than 8 Hz, while theta and delta waves have a lower frequency, less than 8 Hz. The waves with the frequency of less than 8 Hz often are classified as abnormal if they are seen in an awake adult. So now I'd like to tell you something about our corpus, that is the Temple University Hospital EEG data set. This is from where we have gathered all our data for our study. It contains 14 years of clinical EEG data collected at the Temple University Hospital, and it is the world's largest publicly available corpus of clinical EEG data. And these records have been curated and organized by clinicians, and they have been de-identified from the patient. It comprises approximately 17,000 sessions from around 11,000 unique subjects. The reference is given for the same. And the number of sessions per year varied approximately from 1,000 to 2,500 sessions. And this took place over from 2000 to around 14 years after that. The majority of EEG data was sampled at a frequency of 250 Hz. So this is the electronic health record of a patient, and I'm showing it to you. So it contains various things, such as first is the reason for the study, then the clinical history of the patient, the medications the patient is on, a short introduction, any technical difficulties they faced, then the description of the record, that is, that particular EEG session is described, along with any abnormal discharges and seizures, if any. Finally, an impression, and then its clinical correlation is given. So from this, we were able to extract all our data. Average number of sessions were about 1.714 sessions per patient. Total patient IDs were around 15,000, while the total number of sessions was somewhere around 11,000. So now coming to EEG as a potential biomarker and its particular use in ADHD. So what is ADHD? Attention deficit hyperactivity disorder is the most common neurobehavioral disorder of childhood. It is characterized by three characteristic symptoms, that is inattention, impulsivity, and motor hyperactivity. Why is ADHD so important? The estimated number of children ever diagnosed with ADHD, according to a national 2016 parent survey, is 6.1 million. In addition, an estimated 8.7 million adults live with ADHD in the United States, resulting in a total societal excess cost of around $122.8 billion. Compared to other datasets, our study has a much larger sample size of 51 patients of ADHD with a total of 77 sessions of EEG. So I would like to tell you briefly about the theories of ADHD. There are four theories of ADHD: the executive dysfunction theory, the state regulation model, the delay aversion and dual pathway theory, and the dynamic developmental theory. The executive dysfunction theory states that the symptoms of ADHD are caused due to deficits in higher order cognitive processes, such as planning, sequencing, and holding attention to a task. And dopaminergic and noradrenergic neurotransmitter dysfunction are implicated in the same. Second is the state regulation model. The state regulation model is dependent on describing two states of the brain, that is the arousal state and the activation state. The arousal state is a physiological response to input, while activation is a voluntary readiness towards action. And these two are dependent upon the effort allocation. In ADHD patients, it was hypothesized they have a suboptimal arousal or an activation state, and that is resultant because of less effort allocation. This was seen even more in slow tasks, which were assigned to these patients of ADHD, in which the suboptimal arousal and activation state became even more prominent and pronounced. So in such patients, it was seen that less effort was allocated, and as a result, clinically it was seen that children with ADHD showed a higher heart rate variability in the slow condition only, suggesting less effort allocation. So it was hypothesized that children with ADHD have a difficulty in keeping an optimal activation state. Then the next theory was the delay aversion and dual pathway theories. This stated the presence of two circuits, the executive circuit and the reward circuit. The executive circuit is involved in executive dysfunction due to inhibitory deficits in the same, while the reward circuit is based on delay aversion. Delay aversion is that patients of ADHD try to avoid any sort of delay by opting for an immediate reward. And that is what is the delay aversion theory. And this leads to a shortened delay reward gradient. And this is the second pathway or the circuit involved in ADHD. Both these together lead to symptoms of ADHD independent of each other. Then finally, the latest theory is the dynamic developmental theory. It takes all the factors into account. And in this, drugs, genetics, and toxins all can in some way lead to dysfunctioning dopamine system branches, namely the mesocortical, the mesolimbic, and the nigrostriatal branches. Mesocortical dysfunction leads to inattention and poor behavioral disorganization. Deficits in the mesolimbic pathway lead to shorter delay of reinforcement gradient and efficient extinction, and nigrostriatal deficits lead to motor hyperactivity and clumsiness. So this was all about the theories. Now coming to ADHD and EEG. So the first ever finding that was noted in patients of ADHD on EEG was that in patients of ADHD, there was an increased absolute theta wave power and a decrease in absolute beta wave power. We just discussed that in normal adults, theta waves are not seen in an awake state. But in ADHD patients, this absolute power of theta waves is increased. Eventually, when it was noted that theta power is increased and beta power is decreased, so scientists came up with a theta to beta ratio, which was the first marker used for ADHD. And this theta to beta ratio was found to be increased in patients of ADHD. This topographical heat map also represents the same. There's an increased theta power in ADHD patients and a decreased beta power, leading to an overall increase in theta to beta ratio. The second marker was ERP, or an evoked response potential. Evoked response potential is essentially a transient neural dynamic, and this has been explored for distinguishing features of ADHD. Advantage is that they can be linked with specific stages of processing. So in the first graph, as you can see, the raw EEG data has been converted into a graph which is plotting power versus frequency. And as you all know, that theta wave is a low frequency wave, and the power of this theta wave was found to be increased, while the power of a higher frequency beta wave was found to be decreased, as we just discussed. Now, in the second curve is what is representing the ERP. What is seen in an ERP is that a stimulus is given, and 200 milliseconds after the stimulus, the EEG recording is noted. This 200 millisecond recording is called the P200 or the P2 or the P2 reading. This is then plotted in the form of a graph in the form of frequency versus time graph, and the power is represented in the form of a heat map to note the three-dimensional plotting of the power of these different waves noted in an ERP. So this graph essentially summarizes it all. The blue graph represents the controls, while the red graph represents the ADHD patients, and theta power at the central zone was found to be more in ADHD patients, while the P2 amplitude, as discussed, was found to be lower in patients of ADHD as compared to controls. Then in another study by Laszlo, Tombor et al., a significant non-linear association between resting gamma power and age was noted in the lower frequency gamma 1 range, that is the 30 to 39 Hertz phase in ADHD as compared to controls. So as you all can see again, that the blue curve represents the controls, while the red curve represents the ADHD patients. And as age increases, a gap is noted between the two curves. That is, the absolute power of gamma 1 frequency is found to be lower in ADHD patients as compared to controls. Now, talking something about alpha waves, which we have not talked about till now. In a study by Keown et al., it was found that ADHD symptoms were associated with a stronger relative right frontal alpha power. That is, the power of alpha waves was found to be more in one frontal lobe as compared to the other, which is called frontal asymmetry. And in a study by Alperin et al., it was found that the relationship between alpha asymmetry and ADHD depends on the negative affect level. And it was found to be an inverse relation. In patients with ADHD having a high negative affect, it was seen that the alpha asymmetry is less, while in ADHD patients with a low negative affect, high alpha asymmetry was found as compared to controls. So essentially, after we looked up all of these studies, in our ongoing study over our data set, we are trying to compare our findings with the previous studies, and in the process, we are trying to find a biomarker for diagnosis of ADHD. And once we have those findings, we would like to share those findings with you. So these are my acknowledgements, and thank you. If you have any questions, you can go ahead.